It's Dr. Friedman again. Welcome back to week 2 of the Jerusalem Science Contest. This week is devoted to chapters 6 and 7 of your textbook. Chapter 6 is Classification of Biological Organisms, and chapter 7 is entitled Plant Systems. So before I begin, I want to apologize. Last week I told you that I would not be giving you material or rehashing material that was already in your textbook. Unfortunately, I did the lecture before I actually looked at the textbook, so there was some duplication. In the future, that will not be the case. So what I'm going to talk to you about today will be something that's a little bit different, and I'm going to start by showing you some slides. The title of this talk is Phylogenetic Nomenclature, Evolutionary Taxonomy, and Molecular Biology. So it's actually, the first two terms are actually pretty much the same thing. The last one is a little bit different. But we're going to look at various aspects of this. And all these things have to do with something called taxonomy. Taxonomy is really, it's a form of, nomenclature and taxonomy pretty much means, nomenclature has to do with naming things. And taxonomy is kind of a classification system. So in biology, we talk about a group as being a taxon, and the plural of that is taxa. So what we're going to do is we're going to look at why do we even need nomenclature? What's all the fuss about? Well, I'm going to show you on the next slide. And this slide entitled, "Will the Real Gopher Please Stand Up?" It's a little whimsical, but what I wanted to show you is there's actually 3 organisms that are shown here. And most people would look at this and say, well, that's a rat, and that's a turtle, and this is a gopher. So this guy, stand up, you're the real gopher. But in fact, the word gopher is really not a scientific term at all. And if I were in Alabama and I said, I just saw a gopher, most people would assume that I was talking about this creature here, which scientists would call Rattus norvegicus. That's the common rat, the thing that you'd find running around in an alley somewhere. The second creature here, if I were in Florida, people would, in certain parts of Florida, people would also refer to that particular organism as a gopher as well. And this is actually a burrowing terrapin. It's a kind of burrowing turtle. And this particular one is Bopherus polyhemus is the scientific name of that. So we've got 2 different names here, and they actually distinguish between these 2 organisms. And the last one, which in my neck of the woods is what I think of when somebody says a gopher. This is actually a pocket gopher, and a Plains pocket gopher is Geomys burgarius is the scientific name. So we've got 3 different names, scientific names, for 3 different animals, and wherever we would be, anywhere we would be, and we talked about these 3 animals, a scientist would know what we were referring to. But if we just said gopher, even though I think most people would say that's a gopher, but there are places where these things are gophers. So we can't really use that system of nomenclature. It's not very useful to us. So I want to look a little bit at the evolution of biological classification systems. How do these change through time? Well, back in the 1700s, Carolus Linnaeus in 1735 was the first one to actually start naming things, unless you want to say Adam Rishon was really the first one. He was the one that gave names to all the animals, but Linnaeus actually wrote down and categorized perhaps something like, I think it was on the order of 15,000 or maybe 20,000 different species of animals, and he applied Latin terminology to something that was actually descriptive of the animal, and those became the scientific names. Many of these names, or most of these names, are still in use today. So the names that Linnaeus gave in 1735 are still the same names that we give today. But in order to differentiate between the different kinds of creatures, he basically categorized them as either being animal or vegetable. So we have Animalia and Vegetabilia, which are the 2 kingdoms that were in use for well over 100 years. And then we have a new classification that came about in around 1866, and in this case we now have 3 kingdoms. So we still have Animalia people were starting to use the microscope a little bit more. So we have Plantae, which is your Vegetabilia, but we also have under that same category, we also have Protista. Protista, these were microscopic creatures that people were starting to see, and well, they had to put them somewhere, and they didn't really fit nicely into Plantae or Animalia, so a third kingdom was created. And then in 1925, there was a super kingdom that was created called Prokaryota and Eukaryota. So everything, these empires are really super kingdoms. I mean, it's something above a kingdom. So they were using that classification. Then Copeland in 1930, I guess that's 1938, developed a four-kingdom system, which has Monera, Protista, Plantae, and Animalia. So basically, it's the same three-kingdom here, but a new kingdom called Monera was actually added to that. And that was expanded to 5 kingdoms because it was found, this is much later, this is 1969, fungi, which were originally part of Plantae, were taken out, because if you look at the, for instance, if you were to look at the cell wall of a mushroom versus the cell wall of a plant, at this point people were starting to do chemical analysis, and it was found that the plant cell wall was composed of cellulose, but the fungal cell wall was actually composed of chitin, which is the same kind of material that you would find in arthropods and things like insects and crustacea. It forms their exoskeleton, but that's the same thing that you would have in the cell wall of fungi. So that gave us a total of 5 kingdoms. And that was later expanded into 6 kingdoms. And by the way, your book, I said I didn't want to refer too much to the book, but your book actually lists these 5 kingdoms. And it's interesting because Monera, since about 1977, Monera has actually fallen out of use. Nobody uses Monera anymore. It's like when I originally learned the hierarchy of classification, I learned that it was kingdom, phylum, order, family, tribe, genus, and species. Now the tribe, which was the third from the last categorization, nobody uses tribe anymore. You can still find it in the literature, in the older literature, but you won't find it in the modern literature. And it's the same thing when you're looking at this kind of thing. Under the kingdoms, you won't find Monera anymore. Nobody's using that anymore, but it is in your textbook. If I were to ask you a question about it, don't assume that it's actually a current term. It's not really. We don't really use it anymore. So then it was divided up into, Monera was basically divided into Archaebacteria and Eubacteria. And they were kind of combined together in a domain called bacteria. So then we now have 3 domains. We have bacteria, archaea, well, they were actually divided out, and the eukaryota. So these are the prokaryotes. This is actually a division of the prokaryotes, bacteria and archaea, and then eukaryota, that just carries over from here. Cavalier Smith in 1993 devised an 8-kingdom system, which he redevised it as a 6-kingdom system in 98. So here you've got Eubacteria and Archaebacteria, the same things you have here. Archaea, Archozoa, Protozoa, Chromista, Plantae, Fungi, and Animalia. And this 6-kingdom system is pretty much what's in use now from 1998. But I want to show you on the next slide that the domains, let me go back one again. The 3, I show you the 3 domains in 1990, the bacteria, the archaea, and the eukaryota. So the 3 domains here, you can see these are the 3 domains, and I've taken one of the domains, eukaryota, which here is called eukarya. It's the same thing, and shown you how that's divided. That's further divided up into the excavata, which are flagellate protozoa, the amoebozoa, most amoeboids and slime molds, and the ones that we're most interested in for the purposes of this discussion and also for the purposes of this course are the Opisthocanta and the Archaeplastida. The other ones are microscopic things. We don't really care too much about it, but from the point of view of agricultural science, here we've got animals and fungi, both of which have agricultural importance, and here we have land plants. So we pretty much have crops, green algae, which is also of agricultural importance. So you've got these things here, and the animals and the fungi in those classes there. So let me move on here and introduce you to the phylogenetic tree of life. So this is, you could look at this as there's some primordial organism here, and it's giving rise to all these other things. And everything is branching out into trees. And you can see again the three domains, bacteria, archaea, and the eukarya, organized along these three branches. But what this isn't really telling us, it gives us very little information other than who the relatives are on a branch. On this next tree, which is a little bit better organized, another phylogenetic tree, this shows patterns of descent. So we can see that we start out again with the initial organism, and it's actually going to split up. It's going to form other lines. Other lines of descent will come from that. So there's some ancestral organism right here, and it gives rise to three families. The Felidae and the Canidae are the cats and dogs, and the Mustelidae are things like the badger and the otter. So these are the three families, and you can see, and this isn't showing you a complete, it's just showing you one organism. So you've got the Panthera, the Taxidae, the Lutra, and the Canis. So these are four different organisms. Actually, there are five that we're going to be talking about here. So the first one is the leopard, Panther pardus. The next one, the American badger, is Taxidae taxus. And Lutra is the European otter. Canis latrans, these are the two dog-like organisms. Canis latrans is the coyote, and Canis lupus is the wolf. And when we look at this, it's really showing us these patterns of descent, but it's not showing us phenotypic similarity. So if you look at this, closely related organisms may resemble one another, and that could just be due to a common ancestry. But if the lineages evolved at different rates or under different environmental conditions, they may not look at all like one another. So, you've got a common ancestor right here. The leopard would not be included in that, but this would be a common ancestor of both the Mustelidae and the Canidae. And then over here, this would be a common ancestor of the Taxidae and the Lutra. So these two things really don't look very much like one another, but these two along this branch here, they actually do look pretty much like one another. So we can't really make any broad generalizations about phenotypic expression just based on these kinds of trees. Furthermore, branching on a phylogenetic tree doesn't necessarily tell us anything about the absolute age of a particular species. So it's not, this particular one is not really showing us which of these things, the Canis, the Lutra, it doesn't tell us, for instance, whether Lutra or Taxidae taxus arrived on the scene first. So that's a problem with that kind of a tree, but there are other trees, which I'll show you later, where that problem is actually solved. So this slide doesn't tell us, for instance, that the wolf evolved more recently than anything else on here, but it does show us that if we were to look at 2 points of ancestry, and if I call that 1, if I call this point 1 and this point 2, I could even make it 1, 2, and 3, it would definitely show us that 1 preceded 2 and 2 preceded 3. But that's all we can really say about this. What practical application might this have, particularly in agriculture? Well, for one thing, from DNA data, it was found not fairly recently that there were 2 species of wild grasses. and these were identified to be maize's closest living relatives, maize being what we call zea maize, which is actually corn. So the corn plant's closest living relative actually, in terms of its genetics, were actually two wild grasses. And it turns out that these grasses may be useful as reservoirs of desirable genes that that could be transferred, for instance, to cultivated maize using either crossbreeding or genetic engineering. So at some point in time, it may be necessary to actually isolate genes, or we could be doing that right now, isolating genes from these grasses that would give us a superior crop of corn. Now, this next slide here has to do with something called DNA homology. And what homologs are phenotypic, and they could also be genotypic similarities between organisms due to a shared ancestry. And we refer to this as divergent evolution. You can also have what are called analogies, and they're also given the name homoplasies, and we'll see that name a little bit later on. And they demonstrate what we would call convergent evolution. So you can have divergent evolution. A character trait arises, let's say, birds develop wings, for instance, or some progenitor of the bird develops wings. Maybe it's the Archaeopteryx, or I don't know, but some primitive bird develops wings, and then all the other birds that come on after that, they all have wings, and that is a class of divergent evolution. But you can also have convergent evolution, this an analogous type of evolution, where all of the structures, for instance, if I were to look at talking about wings again, and maybe wings wasn't the best example to give you for divergent evolution. We might look at something like the flipper of a whale and the limb of some mammal. That would be a better example of divergent evolution, where there's two, rather than saying bird and bird, two entirely different genera, much different genera, and even different families of organisms, but the structure is the same. Whether you're looking at the whale flipper or whether you're looking at the hand, you see the same kinds of bones. They might be modified a little bit differently, but that's really a case of divergent evolution. But you also have convergent evolution, homoplasies, where if you were to look at, for instance, wings in a bat versus the wing of a bird. I mean, these were two evolutionary events that occurred at two probably entirely different times, and it's just the wing happened to have evolved in two different organisms. It didn't evolve from a common ancestor. That's the point of that. So in this slide, we want to look, this is actually trying to demonstrate that something is homologous. And I'm just giving you a little snippet of DNA here from two organisms. We don't have to identify what they are, but you can see that they both, in these two snippets here, so this could be the primordial organism, the thing that was going to give rise to some other organisms. And then divergence occurs. And what causes that, for instance, might be a deletion of a gene. So one gene, in this particular case, I think we've taken out we've taken out this guanine residue. That's been deleted. And you could also have a genetic insertion. So in this case, the GTA combination was inserted between the T and the C here. So that gave us now CCAT, CAA, GTCC, and now we've got another nucleotide portion here that's 4 base pairs longer than this one. Now, if this is a very, very long molecule of DNA, and you've got this kind of a thing, and you're trying to align these things, it's very, very difficult. It might not be so hard to do it when it's something really short like this, but you might have a lot of these along the chain. So when you want to try to align them and see how close they are to one another, you can use computer-assisted realignment, where the computer is actually able to parse this out into these pieces. So it sees the CCAT, and it puts it here, and then it sees the CA, and puts that over here, and then it sees the AGTCC, puts that over here, and it recognizes the fact that there was an insertion here and a deletion here that gave rise to that. So we can really say that this is a DNA homology, that there is actually a homologous relationship here. Okay, I've got just want to see. There we are. Okay, so this is the cladistics slide, common ancestral classification slide, and on this particular slide, what I'm showing again is here's the progenitor organism, the ancestral organism here, and we've got two different lines here, which we'll just call 1 and 2, and you can see that there are three organisms that are coming out of here. So we've got organism A, which is kind of a direct descent from this, and then there's a split here, another divergence that occurs here, and we get B and C. So if we look at these three organisms, A, B, and C, we say that these are monophyletic. And what monophyletic means is that they're all coming from the same. There is a root organism here, an ancestral organism, and when we have the ancestral organism and all of the other things that are derived from it, we call that grouping a clade, and that's monophyletic. If we look at D, E, and F, on the other hand, what we've got there is we don't have a clade here. It's actually short of a clade, because what we've got here is, in order for this to be a clade, it would have to include this ancestral organism, and it would have to be D, E, F, and G. Since we've actually left G out of it, we say that it's paraphyletic. So that's what that means. And then finally, the last case that we have is where we've got this clade here plus one additional organism that's not part of the clade. So this is called polyphyletic. It could be one or more organisms. So if we took this and two organisms, any two organisms here, that would still be an example of a polyphyletic class. Okay, so now I'm going to establish now something called a character table, and I use this actually to construct a phylogenetic tree. And the way it works is we're going to look at 4 characteristics: the possession of a vertebral column, having hinged jaws, tetraploidy, which means 4 limbs, having an amnion, an amniotic membrane, and having hair. And if an organism has any or all of these characteristics, every time it has a characteristic, we assign it a value of 1. If it misses all of those characteristics, we give it a 0. So the lancelet doesn't have any of those things. It doesn't have a vertebral column, it doesn't have hinged jaws, it doesn't have tetraploidy, amnion, or hair. But the lamprey has a vertebral column. And the bass, this fish, not only has a vertebral column, but it also has hinged jaws. But it doesn't have the other 3 characteristics. The frog has a vertebral column, it has hinged jaws and tetraploidy. The turtle's got 4 of them. It's got, in addition to everything else, it has the amnion, and the leopard has all 5. It's got an amnion, it's got tetraploidy, it's got hinged jaws, a vertebral column, but it also has hair. And we can use this information to actually construct a phylogenetic tree, which we're going to do on the next slide here. And this is the tree for that particular group of organisms. So we could call this a clade if we wanted to. If we say we're going to include this and then everything else in here, that would be a clade. So and this would be called a cladogram. So here we have the actual tree, and it's showing, because of the fact that this didn't have any of the characters, it's the most primitive of the organisms, and it comes straight from here. The lamprey and the bass, because they had a shared characteristic, they're actually, we put the lamprey here next, and then the bass, there was a shared characteristic here, we put that next. There's an additional characteristic for the frog, and then the turtle had 4 of them, and the leopard's got all 5. So that's how we actually put together the cladogram for that. Now, the other thing about a phylogenetic tree, I mentioned before that it really, the way I drew the last trees, all those lengths were kind of proportional to one another. I mean, they weren't proportional to one another, they were all the same length. Let me go back one so you can see what I'm talking about. You know, this length and that length and that length, they're all pretty much the same. So now, what I've done is I've taken different organisms, but in this one, I've created something that's called a temporal phylogenetic tree. I just gave it that name. It has to do with time. Temporal means time. So this one is actually having to do with the fact that in this particular phylogenetic tree, we actually can say something about not only when something happened, it's actually showing us not only when a particular change occurred, but it's also showing us, giving us the same information that we got from the last tree. But we can see that the Drosophila, and we can do this, there's a number of ways that we can do this, but we can just do it by looking at the genes that are present in the organism and see how they've changed over time. Because all of these things share many common genes. So the Drosophila is the most primitive. The lancelet, which we saw in the other slide, a little bit less primitive than that. I mean, it's been around, actually the Drosophila, if you look at this, it actually, this organism was pre-Paleozoic. So that's before 542 million years ago. The Drosophila, or its ancestors, something that probably looked very much like the modern Drosophila, was around at that time. That's a fruit fly, by the way. During the Paleozoic era, the lancelet then was derived, and you can see that in the mid-Paleozoic period, we got the zebrafish, we got the frog, and then the chicken, or birds, they came around in the very late Paleozoic period. At this point right here, 251 million years ago, there was a mass extinction that occurred at this point. So about 90% of all life, all species that lived on the planet Earth were destroyed, and the remaining 10% then had many, almost not quite 200 million years, 180 million years or so to further evolve and form other organisms, and I show a couple of them here. And there was they evolved right after another mass extinction. This is called the sometimes referred to as the KP extinction, or that's what it was. It was the Cambrian Permian extinction, and that occurred about 65.5 million years ago. No one's really sure what caused this. This was probably an asteroid. This has been proposed as either an asteroid or maybe a lot of volcanoes erupting that caused a climate change that killed everything. There have been other theories that have been proposed, but everybody's pretty agreed that this was either an asteroid or perhaps a very large meteor that struck the Earth and caused this extinction. Anyway, after that, this killed the dinosaurs, and then the mammals, which were little things, began to take root, and you had probably the progenitor of the mouse originating quite a bit after that period of time, and then human beings, we've only been around for a few million years. So that actually shows you, it is giving you some information regarding the amount of time that these things took place over. Now here's something called a molecular homoplasy. So what's actually happening here is I'm looking at base pairs here again, and I think there's 24 bases here. You can count them. I'm pretty sure it's 24 bases, but these would be, these are not base pairs here. These are bases from two different organisms. So you've got this little piece of DNA here, and this strand of DNA here. And when we talk about homoplasy, these are the similarities between these organisms. But the reason it's a homoplasy, it's not a structural type of thing. It's not something that was due to convergent evolution. That was due to divergent evolution. This is really what this is. It's actually a coincidental kind of thing. If you look at it, you'll see that the base pairs that align here, there's 3 base pairs that align here, 2 base pairs that align here, and 2 base pairs that align here. which means that there is some sharing common DNA between these two organisms. But if you think about it, since there are only 4 base pairs, there's A, T, C, and G, just randomly, statistically, if you had 24 base pairs, the number of base pairs that would be similar, that would be identical, would probably, on average, be about 25%, or 6 base pairs out of 24. So that's what we're seeing here. So when we look at something like that, this shows shared DNA from 2 totally unrelated organisms, but we would say that only 25% of it is shared, and that's, like I said, that's just what you would get based on there being really no similarity between these things at all. So we would say that this has a 75% genetic difference, and that's what you would get based on just random statistical variation. If we look at this next slide, and here I've got 3 organisms, a human being, a mushroom, and a tulip. And we're going to look at the genetic difference between all 3 of these organisms. So an organism with itself obviously doesn't differ. So human to human is 0. There's a 30% difference in the DNA of a human and the DNA of a mushroom. So it's a little bit more than what you would get just from that random statistical. Well, actually, it's quite a bit better, because it's really telling us that 70% of the DNA. That means that 70% of the DNA is the same. Only 30% is different. So it actually is quite better than just a random type of thing. So it's telling us that there must be some similarity between the human being and the mushroom. If we look at the tulip, that's a 40% genetic difference. So it looks like humans are closer to mushrooms than they are to tulips. So if we look again over here, we'll see that between a mushroom and a tulip, there's also a 40% difference. So what can we do with that information? Well, what we can do is we can establish actually 2 different phylogenetic trees here. And if you look at these percentages that I have here, which are different than the percentages on the other slide, they still add up correctly to the numbers that I want to show you. So if I want to look at the difference between a human and a tulip, what I have to do is I start here, and I have to go here. I get 5% here and 15% here. That's a total of 20%. And then the 20% variance over here, that adds up to the 40% that we saw before. But between a human and a mushroom, what we're going to do, since they're on the same, in this case, we start here rather than here, because this is the point of departure when we're going from tulip to human. We have to start at this point. But here, if we started right here, this would form a clade. This group right here would be a clade. This would be an outlier. This would not be in that clade. So here, what we do is we're just going to start at this point, and then we add 15% here and 15% here, and we get 30%. But there's 2 ways to actually organize this. If you look at it, you could also organize it this way. So this is another way of doing it. We still get the same numbers. So if we look at it from this perspective, we're trying to find the difference between the mushroom, let's say, and the tulip. So we've got 5%, and 10% is 15%, 25%. That gives us the 40% difference. But if we wanted to go here, we would also get a 40% difference going from this point here and here. But if we wanted to go from human being to mushroom, we would start here. We get 5 and 10 and 15, and that would give us the 30. So the question is, which one of these actually is most likely to represent the real tree, the correct tree? And there's 2 principles that we use to establish this. And one of them is called the principle of parsimony, and the other one is called the principle of maximum likelihood. Now, parsimony really means stinginess. So the principle of parsimony is sometimes, there's a scientific principle that's sometimes referred to as Occam's razor. And Willem of Ockham was some kind of a cleric, I think, who lived in the 14th century. And this Latin proverb, I guess you would call it, has been attributed to him. Nonquam ponenda est pluralitas sin necessitat. And what it means is plurality must never be positive. And that's another way of saying you don't really want to, plurality must never be positive without necessity. And that's really another way of saying you don't complicate things beyond the minimum need that is there to complicate them. In other words, don't make things more complicated than they have to be. And so that's the principle of parsimony. So if you look at these things, neither of them really involve more changes. One does not involve more changes than another one does. So in terms of the principle of parsimony, we can't really, if there were different genetic changes that were occurring, if one of them involved fewer genetic changes, or we didn't have to do so much work to get to it, we might say that, well, based on that principle alone, we would assign one or the other. But we can't really do that here. But what we can do is we can use the second principle, and this is the principle of maximum likelihood. And since we make the assumption that the rate of changes of DNA is about the same over periods of time, that the rate of genetic mutation does not increase significantly over long periods of time. By using that principle alone, it would seem more likely that this first grouping is going to be the correct one rather than the second grouping, because this involves a much longer period of time. Here you can see that we have to go to here, and then this is longer than that is. Here these are, we're showing them, when we show them this way, this is really giving us both parsimony and maximum likelihood. So we would say that that would be the correct grouping for that. And last thing I want to talk about are a little bit about evolution here, about the mechanisms of evolution, because all of these genetic changes are occurring because of evolutionary changes. I mean, we talked about a little bit about deletion of genes, insertion of genes. There are things that can occur. Some of them might be environmental, some of them could be chemical, that will cause a base pair mutation, that will cause, I'm going to also talk about something called gene silencing, but there are things that will cause a base pair not only to mutate, but an additional base pair to be inserted, and other things that will cause a base pair, or even multiple base pairs, to be deleted. And if you'll recall, those of you who have biology, you know that when DNA is replicating itself, and it's trying to make an exact copy, if there's an error in replication, that means that there's going to be, that probably, particularly when that thing recopies itself, when that DNA chain recopies itself, it's going to be making multiple copies of the wrong thing. In some cases, if it's a single base pair mutation, there are actually mechanisms within the cell to repair it. If it's a base pair insertion, there are enzymes that will actually delete that inserted base pair. If there's a base pair that's been deleted, there are other enzymes that will actually cause, other mechanisms that will actually cause the base pair to be put back in again. But sometimes those things escape. They escape the repair mechanisms. And when that happens, then you're likely to get some kind of a mutation. But you can also get mutations from chemical and physical environmental things. And I'm going to show you one example. This is the chemical structure. I haven't shown the rest of the molecule, of course, but this part right here is just adenine. This would be the A, the adenine part of it. And if I put on the ribose sugar here, the 2'-deoxyribose sugar, and then I put on some phosphate groups out here, I'd have adenosine monophosphate or adenosine triphosphate, and that would be the thing that would be used to to actually insert that A into the DNA. But in the presence of something like nitrous acid, and by the way, if you eat, it's really not a problem for people that observe kashrut, because they're not eating things, a lot of the things where nitrites are used as preservatives. But nitrites are frequently used as preservatives, and nitrites can also form naturally from nitrates, so it's possible that these things could actually wind up also being in the soil. And it turns out that when adenine is exposed to nitrous oxide, nitrous acid, not nitrous oxide, but the nitrous acid, that adenine will actually, it undergoes a chemical reaction called a diazotization, and then ultimately the amino group here, the NH2, is actually transformed to an OH group, to a hydroxy group. So it's no longer adenine, it's now a different base, a non-natural base, which is called hypoxanthine. Now it turns out that adenine, the base you have here, pairs with thymine. That would be the pyrimidine. But this base here no longer pairs with thymine. It actually pairs with cytosine, the other pyrimidine. So this pairs with a C. So where you'd expect to get a D inserted into the a T inserted into the copy here, instead of getting a T, you're going to get a C once this base pair is modified. So when proteins are made, when RNA is made from the DNA, and we get a messenger RNA, and the message tells us for every 3 residues that we have, that's called a codon, that 3, like an A, T, C, etc. Those combinations, ACG, that would code for a particular amino acid. So once we put the wrong code in there, we're going to get the wrong amino acid, which may mean that we're going to get an inactive enzyme, or we're going to get something that's going to do something entirely different. Those kinds of mutations, if enough of them actually occur, that could actually cause a new species to develop. So that's one possible mechanism of speciation. By the way, this nitrogen is a little bit off here. It actually belongs right at this point here. Same over here. So that's just a little kind of a problem that I had when I was making the slide. I couldn't get the nitrogen to stay exactly where I wanted it. The next slide is going to show another kind of mutation. This is a photochemical mutation, and my oxygen's a little bit off as well. But this is the nitrogenous base. Actually, the one that I've shown here is this would actually be uracil. I should have probably put a methyl group over here. So if I had a CH3 over here, that would be a methyl group, and then this would be thymine. This could also happen in uracil, but it's more likely to happen in thymine with the methyl group. So if I had 2 molecules of this and it was exposed, this symbol here, H nu, stands for ultraviolet light in this particular case. It's some kind of light. It would have to be in the UV range in order for this reaction to take place. And we'd have a methyl group here and a methyl group here. 2 molecules of the thymine would condense to give us this material, which would be called a thymine dimer. This can also happen as well with cytosine, so we could also get a cytosine dimer as well. This is called a 2, in organic chemistry, this is called a 2+2 cycloaddition reaction. And it gives us this 4-membered ring here, which is a cyclobutane ring. And when this happens, so we'd have to have 2 thymine residues that were next to one another in a DNA chain, and it would form this kind of a structure here. Well, it causes a kind of a bulge in the ring, and when the polymerase that's actually reading this comes to this thing, it doesn't know what to make of it. So if this doesn't get repaired, and it can be repaired, but when it's not repaired, it's either going to stop copying, or if it continues to copy, there's a very good chance that it's going to actually add the wrong base, that it's not going to know what to do with this, and it's just going to maybe insert some random base. So this is something, exposure to the sun could actually cause this to happen. So that's another mechanism of chemical mutation. And then there's another I think this is the last slide, just a couple things that actually do occur naturally all the time, and And these are not, this is just part of the normal genetic process, but something called gene silencing. And gene silencing actually has to do with the fact that even though genes may be present, they're not always active. So it was known, it used to be, if you went back maybe 20, 30 years ago, people would say that whatever message is present in a gene, I mean, whatever message is present in the DNA, that really is everything that you need. That really determines all kinds, everything that you're going to get in terms of a phenotypic expression is going to come directly from the genotype, and that's all you really need to worry about. And then now there's a whole area called epigenetics, where it was found that there are other factors besides the genes alone that are important for controlling gene expression. And one factor that I'm showing right here is something called methylation. So this is one process here. So here, this little squiggly line here just represents that, again, this would be attached to a 2'-deoxyribose, and the phosphate groups and other, out here somewhere we would have other, this would be some kind of a polynucleotide here. And there are enzymes that will actually add a methyl group at this position. So this is cytosine. If we methylate cytosine, and cytosine tends to be methylated when it's next to a guanine. If that's going to happen, it's usually these CG combinations that act as a signal for methylation. So that's one mechanism when this methylates that can actually silence the gene. And this double-headed arrow is really showing you that this is a reversible reaction. So when it needs to be silenced, it's methylated. There's another enzyme that will take the methyl group off and then reactivate it again. Another mechanism of gene silencing involves what is called histone protein. So if you look at a DNA molecule, you'll find that the DNA itself is actually coiled around these protein molecules that are called histones. And they actually are organized in little groups of 8. So they're stacked up, these groups of 8. And then the DNA is tightly wound around the histones. Well, the degree of winding and the degree of packing actually has a lot to do with whether or not the histone, in this case I'm showing what's called an acetylation reaction. So adding this CH3CO group here to this amino group is an acetylation. And when this is acetylated, I believe it's wound a little bit more loosely than it would be otherwise. So this actually is going to change whether or not the gene is going to be accessible. So these are just two different mechanisms, natural mechanisms for silencing genes. And you can this can be acetylated, monoacetylated, it could be diacetylated. There's other mechanisms as well. It could be phosphorylated, you could put phosphate groups on here, it could even be methylated. So all these things will actually control, to some extent, whether or not a gene is going to be available or whether it's going to be silenced. And silencing is something that you want to have happen because you don't want to necessarily have, particularly when you've got a gene from both parents, you don't want both of those genes necessarily to be expressed. You might want one of them to be silenced and the other one to not be silenced. So that's the actual natural reason for having this kind of a mechanism. Well, that's about all I have to say on this particular subject. And next week we'll be looking at chapters 8 and 9, I believe, and your exam again will be probably again a 25-question exam. I guess about 6 questions will come from this talk, and the remainder will come from your